Friends, I am Dr. K.D. Mishra and today we will discuss structure and function of hemoglobin, anemia and polycythemia. Hemoglobin is a red pigment and 90% of the dry weight of RBC is contributed by the hemoglobin. It is a chromoprotein which is made up of two parts, the globin which makes up 96% and the heme part which makes about 0.34%. Heme is a metalloporphyrin compound consisting of 4 pyrrole groups. Each molecule of hemoglobin is made up of 4 polypeptide chains, 2 alpha chains and 2 beta chains. Each chain is attached to a heme molecule with Fe2 plus, and each heme molecule can carry 1 molecule of oxygen. Therefore, each molecule of hemoglobin is capable of carrying 4 molecules of oxygen. Therefore, hemoglobin is very important in the transport of oxygen from the respiratory organs to different parts of the body, tissues and cells. Now, hemoglobin is a globular protein with a diameter of 6.4 nanometer. It has a molecular weight of 68,000. One RBC contains as many as 640 million hemoglobin molecules. So, you can imagine if 1 molecule of hemoglobin carries 4 molecules of oxygen, then how much molecules of oxygen are carried by 1 RBC. Hemoglobin consists of 4 polypeptide chains, 2 alpha chains and 2 beta chains and heme groups. The 4 iron atoms are in the ferrous state, that is Fe2 plus. Synthesis of heme takes place in the mitochondria. Now, here you can see the picture of hemoglobin molecule. There are 2 alpha chains and 2 beta chains, and each of these alpha chain and beta chain is attached with a heme molecule, and those heme molecules can combine with 1 molecule of oxygen. The synthesis of hemoglobin begins during the erythropoiesis at a stage called polychromatophil erythroblast stage, and it continues, the synthesis continues until the reticulocyte stage is reached. So, after this stage, the erythrocytes becomes mature. Now, here you can see the stages in the hemoglobin synthesis. In the first step, 2-succinyl coenzyme A combines with 2-glycine forming a pyrrole. Succinyl coenzyme A is formed in the Krebs cycle. Now, these 4 pyrrole, in the second step they form protoporphyrin 9, and protoporphyrin 9 combines with a molecule of iron forming heme. Now, heme plus polypeptide forms hemoglobin chain, either alpha or beta, and such 2 alpha chains and 2 beta chains form a hemoglobin molecule. In the hemoglobin molecule in which there are 2 alpha chains and 2 beta chains, it is called hemoglobin A. Now, properties of hemoglobin. The hemoglobin shows affinity to oxygen. It is a characteristic feature of hemoglobin. It can easily combine with 4 molecules of oxygen, and then it can be easily dissociated into oxygen and hemoglobin back. This combination or this compound, temporary compound with oxygen, is called oxyhemoglobin. In this form, oxygen is loosely attached with the hemoglobin molecule, and when this oxyhemoglobin reaches to the cells and tissues, due to higher concentration of carbon dioxide, dissociation of oxyhemoglobin occurs and oxygen is released to the tissue and the hemoglobin is back into the system. Now, hemoglobin, as it combines with oxygen, it can also combine with other gases. Say for example, it combines with carbon monoxide forming carbon monoxyhemoglobin. It can combine with NO forming nitric oxide hemoglobin. It can also combine with sulfur dioxide forming sulfhemoglobin. CO2 also combines with hemoglobin. When CO2 is transported from tissue to the lungs, some part of it combines with hemoglobin forming carbaminohemoglobin. Now, types of hemoglobin. Hemoglobin is of 2 types. Broadly, it is categorized into 2 types: hemoglobin A, that is found in the adult, and hemoglobin F, which is found in the fetus. In adult, the hemoglobin A, which is composed of 2 alpha chains and 2 beta chains, it constitute 95 to 97% of the total hemoglobin. A very small amount of hemoglobin A is found in the fetus, as it appears in the 8th week of fetal life, but within 6 months of postnatal life, that is after birth, hemoglobin F is replaced totally with adult hemoglobin. Now, second type of hemoglobin is the hemoglobin A2. A very small amount is found in the adult. It has 2 delta chains and 2 alpha chains, that is the chain. There in hemoglobin A, it was 2 alpha chains and 2 beta chains. Here, with 2 alpha chains, there are 2 delta chains. Its normal range in the adult is about 1.5 to 3.5% only. Then the fetal hemoglobin, as we just discussed, HbF, is the major hemoglobin component in the intrauterine life. It has 2 gamma chains with 2 alpha chains. So, there are variations in hemoglobin from the alpha chains. There are combination of delta chain in case of hemoglobin A2, and in case of fetal hemoglobin, there are 2 alpha chains and 2 gamma chains. It accounts for 70 to 90% at term. When the fetus is at term, then at that time it is about 70 to 90%, but then it falls to only 25% in the first month itself and to only 5% during the next 6 months. Fetal hemoglobin may be found in adult in certain anemias like leukemia. Now, there are certain abnormal hemoglobins. There are 4 important hemoglobins that causes diseases. The number 1 is the hemoglobin Barts. It is a tetramer of gamma chains. So, 4 polypeptide gamma chains are there in this hemoglobin Barts. It is observed in case of alpha thalassemia. So, alpha thalassemia is a disease caused by this type of abnormal hemoglobin, which is called as hemoglobin Barts. Then second type of abnormal hemoglobin is the hemoglobin S, shortly it is called as HbS. It is due to mutation in the beta chain of hemoglobin. The glutamic acid is replaced by valine. Now, this disease is called as sickle cell anemia. As you know that the RBCs becomes sickle shaped and their hemoglobin becomes defective. Then hemoglobin C, that is HbC, is produced due to variation in beta chain. It causes mild chronic hemolytic anemia. Another type of hemoglobin, which is a combination, is hemoglobin AS, that is hemoglobin adult and hemoglobin for sickle cell anemia. So, it is observed when there is a gene of adult hemoglobin containing 2 alpha chains and 2 beta chains, and there is a hemoglobin chain gene which has sickle cell anemia gene. So, in this disease a combination of these two is found due to one gene of normal hemoglobin and one gene of sickle cell anemia. Now, normal values of hemoglobin. In normal adults, the hemoglobin ranges between 14 to 80 gram per 100 ml or per deciliter. The average value comes about 15.5 gram per deciliter of blood. In case of female, the percentage of hemoglobin is little less than males. It is about 12 to 16 gram per dl. An average value comes about 14 gram per 100 ml. This variation in the amount of hemoglobin in female is due to the menstrual cycle. As you know, a lot of blood loss occurs in every month during menses. Hemoglobin percentage in case of the newborn, it is higher. As you know, the number of RBC is directly proportional to the amount of hemoglobin present in the blood. So, in case of newborn, the RBC count is very high and therefore, the hemoglobin percentage is also higher, that is about 22 gram per 100 ml. Now, the hemoglobin percentage decreases after birth. You know, as soon as a child takes birth, the hemoglobin percentage gradually decreases and then it attains the adult value, that is about 14 gram per dl. The hemoglobin percentage also decreases with the age, especially after 50 years of age. Now, variations in hemoglobin concentration. Variation in hemoglobin concentration are mainly due to the RBC count. Any condition which affects the number of RBC, it also affects the hemoglobin concentration. As I said earlier, the hemoglobin concentration is directly proportional to the number of RBCs found in the blood. Now, the hemoglobin percentage is highest in the fetus, as just we have seen. It is about 22 gram per deciliter. At birth, it is about 22 to 23 gram. It falls below normal at the end of third month due to iron deficiency. As you know, during the first few months, the baby is fed with the mother's milk, in which there is a deficiency of iron. So, therefore, the hemoglobin percentage, it falls during that period. At the age of 10 years, it becomes round about 12.5 gram per deciliter, and then it is gradually raised to the normal value of 13-14 gram per 100 ml. Now, hemoglobin percentage varies with the sex, as we have seen just now. In case of female, it is about 13.7. It is little higher in case of males, it is about 15.8 gram percentage. Then there are diurnal variations also in hemoglobin percentage. As you know, we have seen that in case of RBC count, the RBC count is maximum during during evening hours when metabolic rate is higher, and it is minimum during morning hours after rest. Similar variation is seen here. 10% diurnal variation is found in the hemoglobin percentage, being lowest in the morning and highest in the evening. Then certain conditions are there which increases the RBC count, also increases the amount of hemoglobin in the blood, such as excitement, exercise, adrenaline secretion. These conditions causes increase in the hemoglobin percentage. Then at high altitude, the hemoglobin percentage increases, because as we move on the high altitude, there is a deficiency of oxygen in the air. So, demand of oxygen increases, and to meet out that, number of RBCs are increased, and at the same time the amount of hemoglobin increases so as to meet out the oxygen demand of the tissue. Now, decrease in hemoglobin percentage is seen in case of anemias. When there is anemia, there is less number of blood cells, or even if the number of blood cell is normal, then also the decrease in hemoglobin concentration can occur, that is called anemia. It can occur due to hemodilution. Hemodilution means dilution of blood, and the volume of blood increases, that is called as hypervolemia. So, if the volume of blood increases, then the density of RBC will decrease, and thereby the amount of hemoglobin will be decreased, as we can see in the event of hypersecretion of ADH. You know, ADH is a hormone which is secreted from the posterior pituitary, and it has a function that it maintains the osmoregulation, thereby increasing the absorption of water from the kidney. So, if there is hypersecretion of ADH, it will increase the volume of blood by dilution, and thereby it will decrease the concentration of hemoglobin in blood. Now, increase in HV percentage is observed in conditions of hemoconcentration. Conversely, if the blood becomes concentrated, then the percentage of hemoglobin will increase. As you see, in any case, when a fluid is lost from the body or water content is lost from the body, then the blood becomes more and more thicker and dense, and in that case the hemoglobin percentage will increase. Then in case of hypoxia, that means the deficiency of oxygen in the tissue, like in case of congenital heart diseases, lung disease like emphysema, where lungs do not work properly and oxygen is not supplied properly to the tissue, or in such cases like polycythemia, in which the RBC count increases abnormally, the hemoglobin percentage increases. So, these are the conditions when the hemoglobin percentage increases. Now, the hemoglobin forms certain complexes with gases, as we have just now discussed that the oxyhemoglobin is a temporary complex made with oxygen. Each hemoglobin molecule makes a complex with 4 molecules of oxygen. Then this oxyhemoglobin is supplied through the blood to different organs, and there it is disintegrated into hemoglobin and oxygen. Oxygen is being utilized by the tissue, and deoxygenated hemoglobin, sometimes it is called as reduced hemoglobin, is returned back. Now, second one is carbaminohemoglobin. You know, carbon dioxide is also transported by hemoglobin. Actually, majority or maximum carbon dioxide is transported in the form of bicarbonate ions, but some amount of carbon dioxide combines with hemoglobin, forming a temporary compound that is called as carbaminohemoglobin. It is disintegrated then into carbon dioxide and hemoglobin back. Another and very important is carboxyhemoglobin or carbon monoxyhemoglobin. You know, carbon monoxide, which is by and large produced in the automobile exhaust, it has a affinity to hemoglobin, which is 200 times more than oxygen. So, if carbon monoxide is available, then preferably it will combine with carbon monoxide rather than oxygen, forming carboxyhemoglobin or carbomonoxyhemoglobin. It is a irreversible reaction. So, that hemoglobin is not available for making oxyhemoglobin with oxygen. Now, some other complexes with hemoglobin, one is that methemoglobin. In that hemoglobin, the iron is oxidized from ferrous state to ferric state, and it is a stable compound which is chocolate brown in color. It cannot combine with oxygen because it is a irreversible reaction. Methemoglobin oxygen carrying capacity of blood is reduced because that hemoglobin is not available for making oxyhemoglobin. The second one is sulfhemoglobin. It is due to combination of hemoglobin with sulfur. Its oxygen carrying capacity is 100 times lower than normal hemoglobin. Sulfur is attached to porphyrin ring and therefore it is irreversible reaction. Another is nitric oxide hemoglobin. Hv combines with nitric oxide. This is called as nitric oxide poisoning. This is also a irreversible reaction. Now coming on to the functions of hemoglobin, as we have just discussed that the main function of hemoglobin is the transport of gases. It transports oxygen and carbon dioxide both. It carries oxygen from lung to the tissues. It also carries carbon dioxide from tissue to the lungs. Now Hb acts as a buffer. You know, when the carbon dioxide enters into RBC, it forms carbonic acid, that is H2CO3, and this H2CO3 dissociates to form H ions and H3O3 ions. These HCO3 ions, they come out of RBC in lieu of a chloride ion. So this is called chloride shift. By this way, the hemoglobin serves as a buffer system, and during the elimination of carbon dioxide and HCO3, it acts for balancing the acid-base buffer system in the blood plasma. Now estimation of hemoglobin. You know, estimation of hemoglobin is one of the basic priorities of medical examination. During the routine investigation, when the blood is tested for various parameters, one of the very important parameter is the hemoglobin percentage. In routine, for estimation of hemoglobin percentage, a equipment is used which is called Sally's hemometer or hemoglobinometer. You know, this is a sort of box, a comparator box is there, in which there are two colored glasses which are used for reference, and there is a middle transparent glass in which you can put the sample tube. The sample is collected through the hemoglobin tube, and then it is put in the tube and mixed with N/10 HCl, this one. Now this N/10 HCl reacts with hemoglobin, forming acid hemoglobin. This acid hemoglobin is brick red in color. Then we mix distilled water into it while continuously stirring it. till the color of the central tube in which sample is there matches with the standard glass plate color, and then this tube is graduated so that you can read the hemoglobin percentage on this tube. The other methods are also used for this. One is automated hemoglobinometry. There are automatic meters in which you take the sample, add some reagents into it, and then it is put in the hemoglobinometer, and the reading is taken. Another is spectrophotometry, which also you measure the change in the color of the sample after adding reagents into it. Here you can see the picture of hemometer or hemoglobinometer, that is Sally's hemometer, in which there is a comparator box, this one, there is a sample tube here, there is a pipette. Now in the pipette we take a sample of blood, then it is mixed with N/10 HCl, a brick red color appears, then on both the sides of this sample tube there are colored plates. So after adding distilled water into it and stirring continuously with the help of a glass rod, we match the color of this sample tube, and then the reading is taken on the tube, which is a graduated tube, to see the percentage of hemoglobin in blood. Now, critical values of hemoglobin when the blood transfusion is required. You know, the blood transfusion is not recommended as long as the hemoglobin percentage is up to 8 gram per 100 ml and the hematocrit value is above 24%. If the hemoglobin percentage falls less than 5 gram per 100 ml, it is considered critical and the patient is need to be given a blood transfusion. But in case of older patients, the blood transfusion, because you know with the old age the percentage of hemoglobin decreases and the process of erythropoiesis, that is formation of RBC, also decreases. So in case of old patients, if the hemoglobin percentage is less than 10 gram per 100 ml, a blood transfusion is planned. Now certain conditions when increase or decrease in hemoglobin percentage is observed. One is polycythemia, we have already discussed. If the number of RBC increases, accordingly the percentage of hemoglobin increases. Then second is the congestive heart failure. In case of heart failure, the heart is unable to supply the blood to different organs of the body. A hypoxia is created, and due to hypoxia, the production of RBC increases and the concentration of hemoglobin increases. In a disease called COPD, that is chronic obstructive pulmonary disease, in which lungs do not work properly and oxygen is not supplied properly to the organs, then also the percentage of hemoglobin increases. After vigorous exercise, when you are in exercise, your need for oxygen increases and therefore the number of RBC increases, and so the hemoglobin percentage also increases. In case of hemoconcentration, as in case of diarrhea, dysentery, as in case of burns. In case of burn, lot of evaporation of fluids occurs from the skin surface. So, in that case, when the blood becomes concentrated or thick, or the density of blood increases, in that case the percentage of hemoglobin increases. Now, here are the cases in which the percentage of hemoglobin decreases. One is in the case of anemia. In case of anemia like aplastic anemia, when the formation of blood cells in bone marrow is affected, it is called aplastic anemia. In case of hemolytic anemia, when the blood cells are destroyed, in case of iron deficiency anemia and pernicious anemia, when the hemoglobin synthesis does not occur properly due to deficiency of iron, in those cases the hemoglobin percentage decreases. In case of thalassemia, it decreases. In case of various types of malignancies, the cancers, in those cases also the hemoglobin percentage decreases. In certain cases of autoimmune diseases, hemoglobin percentage decreases. In case of dietary deficiency, especially deficiency of iron, which is necessary for the synthesis of heme, deficiency of vitamin B12 and folic acid, which are necessary for the formation of RBC and maturation of RBC and synthesis of hemoglobin in RBC. So, in all these cases, hemoglobin percentage decreases. Now, coming on to the second part of this lecture, that is anemia, that is deficiency of blood, and polycythemia, that is increase in the number of RBC, increase in the RBC count. Now, these are the two types of abnormalities in hemoglobin concentration. When the hemoglobin concentration decreases in blood, it is called anemia, and when it is increases, it is called polycythemia. In anemia, usually it is expressed as hemoglobin percentage. So, hemoglobin is estimated in the blood, and if it is less, then it is considered as anemic condition, because it severely affects the oxygen carrying capacity of the blood. Conversely, in case of polycythemia, in which hemoglobin percentage increases, but polycythemia is usually expressed as hematocrit value rather than hemoglobin percentage. As in this case, the red cell mass, that is the counting of RBC and the viscosity of blood increases. So, for the diagnosis of these two abnormalities, two different parameters are used. In case of anemia, for diagnostic anemia, the percentage of hemoglobin is estimated, and in case of polycythemia, the RBC count and hematocrit value is important. Now, anemia just I explained that anemia is any type of deficiency of blood. It is normally studied as the decrease in hemoglobin percentage or decrease in the RBC count. It is commonly estimated by hemoglobin content of the blood. That is the main criteria for diagnosing the anemia. Hematocrit value, packed sac volume, is also considered. In clinical practice, usually detected by observing paleness in the lower palpebral conjunctiva and at the nail beds. So, if these are whitish or pale in color, that means there is a deficiency of anemia. That is the primary observation a doctor made before going for blood investigation. Now, these are the criterias for anemia. In case of male, if the hemoglobin percentage is less than 13.5 gram per 100 ml, then it is considered anemic condition. In case of female, it is little less, it is 11.5 gram per 100 ml. In case of those who are in pubertic age, this is 11.0 gram per deciliter. In case of newborn, if it is less than 14 gram, it is called anemic. It is taken as a lower limit because of the high hemoglobin. Now, another criteria for diagnosing anemia is the hematocrit value. If it is less than 42% in male, if it is less than 37% in female, it is considered anemic condition. In broad sense, anemia is the blood's inability to supply adequate oxygen to the tissue for proper metabolism. If proper oxygen is not supplied to the tissue, then it should be because of the decrease in hemoglobin concentration. Now, anemia is broadly classified into two categories. One is on the basis of morphology of RBC, whether RBCs are abnormal in shape and size, whether they are large, that is called as macrocyte or megaloblast, whether they are sickle shaped, whether they are smaller, that is called as microcytic anemia, and so on. Or second one is etiologically, that is, if there is a blood loss due to either decreased production of RBC or deficient erythropoiesis, blood cells are not properly formed in the bone marrow, or there is an increased destruction of RBC due to some region, it is due to excessive hemolysis of RBC, then the anemia is observed. Now, morphological classification of anemia. If the RBC remains smaller in size than normal, it is called hypochromic microcytic anemia. That means microcytic means they are smaller in size, hyperchromic means they contain less amount of hemoglobin. So, if the size of the RBC is less than 80 FL, that is femtoliter, then it is called as hypochromic microcytic anemia. Then as you know that hemoglobin contains 4 heme groups. For that synthesis, iron is needed. So, if there is a iron deficiency, anemia is caused. It is called as iron deficiency anemia. The main causes of iron deficiency anemia are in case of chronic bleeding, If bleeding occurs from some part of the body, maybe from the parts of alimentary canal, from lungs, from colon, then the anemic conditions appear, or in case of hookworms, which is a helminthic parasite, the deficiency of blood is seen. Then in case of thalassemia, thalassemia is a genetic disease. Formation of globin is defective, in which synthesis of hemoglobin polypeptide chain is decreased in alpha and beta thalassemia. Respective hemoglobin chains is affected. In case of alpha thalassemia, alpha chain is defective. In case of beta thalassemia, beta chain is defective, and due to that deficiency of blood occurs. Now, second one is called normochromic normocytic anemia. Even if the size of the RBC is normal, even if the amount of hemoglobin is normal in the RBC, then also in certain conditions anemia occurs. This is called as normocytic normochromic anemia. The size and HB concentration of RBC is normal. Its size is 80 to 100 FL. It is usually occur in following conditions. One is due to hemorrhage, if there is a lot of blood loss, hemolytic anemia, in case of blood cells are destroyed in large numbers, in case of impaired production of RBCs in bone marrow, as we see in case of aplastic anemia, when in bone marrow the production of RBC and other cells is decreased. Now, the third one is macrocytic or megaloblastic anemia. In this case, the size of the blood cells, RBCs, remains larger. As you know, the RBCs are produced from a stem cell stage, we call it erythroblast stage, and then it proceeds into various stages where its size gradually reduces and hemoglobin synthesis increases. But in certain cases, when there is a deficiency of vitamin B12 and folic acid, the RBC do not mature, they remain in a stage which is larger in size and nucleated. So, when these cells passes through the capillaries of spleen, they are destroyed very easily, causing anemic conditions. Here is a term we have used, MCV, that is mean corpuscular volume, that is the volume of RBC. The MCV is the index of the size of RBC. Its average volume of an RBC is expressed in terms of FL, that is femtoliters. 1 femtoliter is equal to 10 to the power 5 liter. The value of MCV is normal in adult. It varies between 78 to 96 FL. So, if it is more than that, more than 100, it is called as macrocytic anemia, and if it is less than that, it is called microcytic anemia. Then etiological classification of anemia. Number one is blood loss anemia. anemia caused due to hemorrhage or blood loss. The body replaces the fluid portion of blood, that is plasma, within 1 to 3 days. So, the concentration of hemoglobin becomes lower, it is decreased as the RBCs are not produced to that number to compensate the loss of blood. The RBC concentration becomes normal in the coming 3 to 6 weeks. So, the hemoglobin concentration is back to the normal stage within 3 to 6 weeks if a second hemorrhage does not occur. Now, chronic hemorrhage. Chronic hemorrhage, that means some kind of chronic disease in which a regular hemorrhage occurs, maybe in a small amount, but it occurs for a very long time. So, in case of gastrointestinal disorders, diseases, in case of certain genital diseases, urinary diseases, in case of respiratory diseases, such type of blood loss is observed as the gastrointestinal blood loss seen in case of peptic ulcers. There are ulcers in the stomach called peptic ulcers. There are hemorrhoids from where the blood loss is regularly takes place. In case of hernia, in case of carcinoma of stomach and colon, then ulcerative colitis. In all these conditions, regular blood loss occurs, so causing anemic conditions. Similarly, in case of certain respiratory diseases also, blood loss occurs sometime, like in case of pulmonary tuberculosis, TB, then bronchogenic carcinoma. In these two conditions, anemic conditions appear. In case of female, this is a very common problem in case of female. With the advancing age in the uterus of female develops some fibrous tissues called fibroids, due to which the menstrual cycle becomes irregular and the amount of blood loss becomes very much, causing anemic condition in the females, especially those of age 45, 50 and above. Then aplastic anemia, as just described, that if the formation of RBC, that is production of RBC, is affected in bone marrow, it is called aplastic anemia. Now, it is seen in various conditions. It is also called as bone marrow aplasia. It is seen in case of chemotherapy, it is seen in radiation therapy given in case of cancer treatment. These radiation and the chemotoxicants, they damage the stem cells in the bone marrow and thereby reducing the production of blood cells. Then there are certain chemicals also causes such effect, like benzene, DDT, certain other drugs like anti-cancer drugs, antibiotics, anti-inflammatory drugs. These are also reasons for aplastic anemia, as they affect the stem cells, that is blood forming cells in the bone marrow. The third one is hemolytic anemia. That means if the destruction of RBC is more than the production of RBC, then it is called as hemolytic anemia. It can be due to some hereditary abnormalities, abnormality in the genes. RBC becomes fragile, and when they pass from the spleen, they are destroyed. In such cases, RBC count may be normal or more, but their lifespan is reduced, because when they pass from the spleen, because of being fragile, they are lysed or they are destroyed, and the number of RBC destroyed is faster than they are produced. This causes anemic condition. Now, types of hemolytic anemia. The first one is hereditary spherocytosis. It is a genetic, autosomal, dominant disorder in which the RBCs becomes smaller and spherical. So, when they pass through the spleen, they are destroyed, they become more fragile, and in this case the spleen becomes enlarged. There are symptoms of jaundice, there are symptoms of anemia. In such cases, the spleen is removed, a procedure, a surgical procedure called as splenectomy. So, the destruction of RBCs is checked to some extent. Second one is sickle cell anemia. As we have already discussed, that due to this hereditary disease, there is a point mutation in case of beta chain of hemoglobin. Glutamic acid is replaced by valine. Such defective hemoglobin is called as HBS. At the same time, the RBCs, the shape of the RBC also changes to sickle cell. So, such RBCs do not have proper amount of hemoglobin, and the sickle cells are destroyed easily. So, this is called as sickle cell anemia. Sometimes the hemolysis is so much that it can cause death. Another case is the erythroblastosis fetalis, in which the fetal RBCs are hemolyzed. This is because of the fact that there is a incompetency of Rh blood group between mother and the fetus. If the mother is Rh negative and the fetus is Rh positive, then in that case, if the mother is already sensitized for Rh and if it has Rh antibodies, so these Rh antibodies can pass through the placental barrier into the fetal circulation, and these antibodies destroys the RBCs of fetus. So, this is called as erythroblastosis fetalis. Iron is a key element for synthesis of hemoglobin, because a hemoglobin molecule has got 4 heme molecule formed of iron pigment. So, if there is a deficiency of iron, it is called as iron deficiency of anemia, popularly called as IDA. In IDA, RBCs may become microcytic and they may contain less amount of hemoglobin, therefore, it is called as microcytic hypochromic anemia. This type of iron deficiency anemia is very commonly observed in the developing countries because of the dietary deficiencies of minerals. Iron deficiency occurs and this causes iron deficiency anemia. It is especially seen during pregnancy when the demand of iron is more. It is for mother as well as the fetus, and if that much amount of iron is not supplemented in the diet, then it causes anemia. Then during the lactation period also, in the childhood also, when the child is mainly fed with the mother's milk, does not contain much amount of iron. So, these are the causes of iron deficiency anemia, IDA. Now, one of the severe effects of anemia is that due to less number of RBC, when the RBC count falls as a cause of anemia, then the viscosity of blood falls as low as 1.5 as compared to 3. So, it becomes just half than the normal. So, what happens as a result? The amount of blood returned to the heart contains more fluid than RBCs. This further affects the tissue hypoxia, because if less number of RBCs are there in the blood, it will generate tissue hypoxia, and when tissue hypoxia is there, then the heart will be stimulated to pump more and more amount of blood, that is increase in the cardiac output. So, this goes on like this, and if such a person does some exercise, then this can be fatal and it can even result in cardiac failure. Now, polycythemia, as we discussed earlier, an increase in the amount of hemoglobin in blood is called polycythemia. It can be categorized into two categories. Number one is primary polycythemia and number two is the secondary polycythemia. The primary polycythemia, also called as polycythemia vera, it is a serious condition, it is a pathological condition in which the RBC count increases from 5 million per cubic millimeter to 7 to 8 million per cubic millimeter, and the hematocrit value increases to 60 to 70% as compared to the normal value of 40 to 45%. It is caused due to some kind of genetic aberration in hematocytoblast cells. That means that those cells which forms RBCs, the RBC stem cells, which continues to produce these hemocytoblasts, continues to produce erythroid series, granulocyte series, megakaryocyte series in uncontrolled way. So, these hemocytoblasts, when there is some genetic disorder, they produce uncontrolled number of RBC, WBCs, megakaryocytes, which result in increase in all the formed elements of blood, including RBC. The treatment is, because all the formed elements increases in the blood, so viscosity of blood increases. For controlling that, nearly 500 ml of blood from the patient is withdrawn every 2 to 4 days. This is called phlebotomy. Or certain myelosuppressive drugs are given, which suppresses the activity of hemocytoblast, thereby reducing the production of formed elements of blood, including RBCs. Now, secondary polycythemia. Secondary polycythemia is caused due to hypersecretion of erythropoietin. As you know, erythropoietin is a hormone which is secreted from the erythropoietic tissue of kidney in response to tissue hypoxia, that is deficiency of oxygen in the blood. So, if hypersecretion of erythropoietin occurs, then there is an increase in the number of red blood cells. It increases to 6 to 7 million per cubic millimeter. This condition occurs in certain cases like tissue hypoxia. If hypoxia is there, more and more amount of erythropoietin will be secreted. At high altitudes also, when there is a deficiency of oxygen in the air, more amount of erythropoietin is secreted. There is some breathing problem, lesser delivery of oxygen to the tissue, as in the case of certain cardiac failure. So, when there is a tissue hypoxia, erythropoietin is secreted in abnormal quantities, and that produces abnormal number of RBCs. It is called as secondary polycythemia. Now, this is called as physiological polycythemia. You know, when one goes on high altitude due to less amount of oxygen in the air, hypoxic conditions develop and more amount of erythropoietin is secreted. As I just said, the when a high altitude of 14,000 to 17,000 is reached, hypoxia appears in the tissue, and that increases the production of RBC from the bone marrow, and the number of RBC becomes 6 to 7 million per cubic millimeter. So, this is called as physiological polycythemia. That's all for today. Thanks.